biomechanics lab principle I looked at was the influence of fatigue on muscle activation of forearm flexors and extensors as seen through EMG amplitudes. Research has shown a linear relationship between time to fatigue and EMG amplitudes during isometric forearm flexor contractions. This research showed that as forearm flexors fatigue, the amplitude of action potentials increased in the muscle. The question I asked in this experiment was, in healthy individuals with full forearm function, will induced fatigue cause an increase in EMG amplitude? I hypothesized that forearm flexors and extensors will show an increased amplitude as time to fatigue is reached. I performed tests on 8 subjects, 6 males and 2 females with a mean age of 23.38 plus or minus 1.51 years. Subjects were volunteers from the Regis DPT class of 2019. Exclusion criteria for selection of our subjects was forearm pain within 6 months, requiring medical attention or activity reductions, history of arm or forearm surgery, and latex allergy or adhesive sensitivity. 6 electrodes were placed on each subject, 2 on the forearm flexors, 2 on the forearm extensors, and 2 on the biceps brachii as grounds. The subjects then performed maximal voluntary contraction by squeezing the hand grip dynamometer as hard as possible. For the test of fatigue, 25% of the maximum force was determined and used to induce fatigue. The subjects held the hand grip dynamometer at 25% force until fatigue or 300 seconds was reached. I then mathematically turned the recorded EMG amplitudes into percent standard amplitudes and determined the mean and standard deviation for all 8 subjects combined. Results of this test were not similar to what previous research has shown. The mean amplitudes of the extensors and flexors were both static throughout the test or showed a slight drop. The standard deviations were also as big or bigger than the means, which caused no significant difference to be seen. These results could be due to the fact that the sample size was so small. If more subjects were tested, change in amplitude might have been seen. The percent standard mean showed a nonlinear relationship as fatigue was reached. Other research has shown a linear relationship with a positive slope as fatigue was reached. This nonlinear amplitude could be attributed to motor unit synchronization or amplitude cancellation. This could also be due to the idea that as fatigue increases, muscles could be receiving less blood or metabolic products. Previous research has shown that fatigue can cause a decrease in blood flow to contracting muscles, which could cause an oxygen deficit, calcium deficit, or buildup of lactate that would all have an effect on muscle activity. These could cause neuromuscular activity to become less effective and thus cause the amplitude as recorded by EMG to look different than how other research has shown. These results rejected my hypothesis as there was no increase in amplitude as fatigue set in. The test is clinically important because it can be used to determine if the patient has a potential neuromuscular problem or physiological problem that is affecting the muscles. This information can allow the PT to determine what the best course of treatment is for the patient and develop goals that